Welcome students to this module of e-learning. I am Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present this module to the third year students of Botany, Semester 6, BOC 109, Molecular Biology and Genetic Engineering. The name of the module, DNA Isolation, Sanger and Coulson's method, belongs to Recombinant DNA technology. The outline of the module is DNA isolation and sequencing, the purpose of DNA sequencing, requirements and procedure of Sanger's method of sequencing. The learning outcomes. Once the student completes this module, the student will be able to explain the terms DNA isolation and sequencing, describe the steps involved in the Sanger's method of sequencing, its uses, limitations, advantages, and disadvantages. Dear students, if one wants to do DNA sequencing, you need to isolate the DNA in the pure form. And this is the procedure which I will be showing you for DNA isolation in the form of a flowchart. The plant tissue, we do cell lysis for that by physical or chemical treatment, followed by treatment with detergent or separation of DNA by specific adsorption to membranes. We will be able to remove the membrane lipids, followed by protease treatment. With protease treatment, the protein denaturation and removal will be achieved, followed by removal of other cellular contaminants. Later, RNase treatment to remove the RNA. And finally, you will get the purified DNA, which we'll be using for the sequencing. Now, what is this DNA sequencing, students? It is the process of determining the sequence or the order of the nucleotide bases. And which are the nucleotide bases, students? The adenine, thymine, cytosine, guanine. And in a particular piece or stretch of DNA or a section of DNA. Now, what is the need for doing DNA sequencing? You have a purpose, student, and that purpose is to deciphering the code of life, for detecting mutations, for typing microorganisms, identifying human halotypes, and designating polymorphisms. Now, in the year 1977, students, there were two separate methods for large-scale sequencing, and these two were devised by separate scientists. The first method was by chemical cleavage method by Maximin Gilbert, whereas the second method was an enzymatic chain termination method by Frederick Sanger. In this module, we will learn about Sanger's method, students. And this method is the most common approach to sequencing DNA. And if you see on your right-hand side, it is Frederick Sanger. He is responsible. He is the scientist who invented this Sanger's method of sequencing in the year 1977, for which he received the Nobel Prize in the year 1980. Now, Sanger's method of DNA sequencing is also called as chain termination reaction, dideoxy method, or Sanger's method, students, named after the inventor Frederick Sanger. It can be known by any of these three names, students. Now, the principle underlying this method, that is Sanger's method, is based on the selective incorporation of chain-terminating dideoxy nucleotide. And what are these dideoxy nucleotides, students? They are fluorescent chain-terminating nucleotides, which are lacking the 3 OH ends. Now, these are added by DNA polymerase during in vitro DNA replication. There is enzymatic synthesis of the complementary strand of DNA. Chain terminates at specific nucleotides, that is the dideoxy nucleotide, then followed by gel electrophoresis and to reading the DNA sequence. Now, let's understand the difference between what is a deoxynucleotide and a dideoxynucleotide. The normal deoxynucleotide triphosphate has the OH group at the 3' end, whereas the 3-OH group, which is necessary for the formation of the phosphodiester bond, is missing in dideoxynucleotide triphosphate. This is the main difference, students. The requirements for this procedure: it will be performed in 4 separate test tubes, and each test tubes will have a single-stranded DNA to be sampled to be sequenced, then a heat-stable DNA polymerase, a primer, 4-deoxynucleotide triphosphate, adenine, thymine, cytosine, guanine, and a very small amounts of the 4-di-labeled chain-terminating dideoxynucleotide triphosphates. Now, the steps involved in sequencing. First, we need to denature the template DNA to separate the 2 strands. Now, once we have done that, we cool the strands. The template strand is hybridized to the primer. This is the primer, students, and it is hybridized to the template strand. Okay. Following this, the temperature is raised again. DLA polymerase acts now, does its function. What is its function? To add deoxynucleotides to the new strand, to the template strand, starting from the primer. Now, DNA polymerase starts putting at random deoxynucleotide triphosphate, and the primer gets extended. Now, this synthesis continues until the template strand or in the test tube, it come across a dideoxynucleotide triphosphate. And at that point, the chain elongation ceases, because at this point, there is an incorporation of a dideoxynucleotide. Now, this dideoxynucleotide, students, I already told you, it lacks the 3-OH group. Now, this OH group is very important to form the phosphodiester bond. Now, once it lacks this, it prevents any further addition of the next nucleotide, and the strand breaks there. Now, if you keep a proper ratio of the deoxynucleotide and a dideoxynucleotide in the test tube, the chain will terminate throughout the length of the template. Now, it's repeated a number of cycles, and this tube now will contain a mixture of terminated fragments of different lengths, which will be labeled with di, indicating that point where the dideoxynucleotide is added. Now, fragments from each of these 4 test tube reaction mixtures are denatured, make them single-stranded, place them in 4 separate gel lanes, and separate them by size through gel electrophoresis. This is how test tube number 1, 2, 3, 4. We have added a dideoxy ATP here. It will stop at A, whereas dideoxy CTP will stop wherever C is added, where it is added, and such strands are created. Now, when you run these fragments in a polyacrylamide gel electrophoresis, the short fragments will move fastly because they run through the pores the gel, crosses first. They finish the line first, followed by the next smallest, while the long fragments move slowly. Now, these separated fragments are then visualized by autoradiography. Now, let's see, students, how the autoradiogram looks like. This is the autoradiogram of the sample by Sanger's method of sequencing. We have these fragments separated according to the sizes, if you see. And this is how it is: test tube number 1, in test tube lane 1, lane 2, lane 3, lane 4, wherein you have added a particular dideoxy here and its strand stops here. Now, fragments are to be read on this autoradiogram from bottom to the top, wherein it is the 5' end to the 3' end. So, the sequence will be read like this. So, it will be C, C, A, T, C, G, T, T, G, A. Okay. So, this is how you read the sequence. C is showing in this column. So, it's a C. C is showing. So, like this, you will mark it and you will read the sequence. Now, the uses and limitations of this method. Its high quality sequence for relatively long stretches of DNA can be attained. Sequence individual pieces of DNA such as bacterial plasmids or DNA copied in PCR. This method, though expensive, is inefficient for large-scale projects. such as sequencing of entire genome and metagenome. So the advantages of this method is this, students, is the most popular method, simpler, quicker, allowing large input. Within an hour, primer annealing and sequencing reactions can be completed. Allows scientists to determine genome sequence and identify the genes causing genetic diseases. If they have advantages, students, they also have disadvantages. They are yielding of poor results owing to secondary structure in the DNA, as sometimes DNA polymerase terminates the chain elongation prematurely, beforehand. The sequence is obtained not from original DNA. We are seeing the sequence from an enzymatic copy, so there is a chance of incorporation of wrong bases. So students, you have understood the method of Sanger's method of sequencing, which utilizes dideoxynucleotides to terminate and get the fragments and then read the sequence by autoradiogram. These are the references, students. These are the online references you can also see on YouTube. And thank you, students, for a patient hearing.